Thank you, Rossella, and good afternoon. Posso avere la prima diapositiva? Yeah. Okay, it's great to have this opportunity to share with you our project and some preliminary, but we feel promising results. So multiple sclerosis is the most frequent and important demyelinating disease of the central nervous system. It is a multifactorial disease with an autoimmune pathogenesis. It involves mainly young adults, particularly females, in the most active and productive period of their lives. And it has a chronic course, and if not treated, it can lead to progressive physical and cognitive disability with high societal costs. Is in fact one of the most important causes of neurological disability in young adults. Italy is considered a high-risk area for multiple sclerosis, and it is estimated that 100,000 people live with the disease in our country. So over the past 2 decades, the therapeutic landscape of the disease has dramatically increased and improved with more than 20 molecules approved in the market. At the same time and in parallel, the diagnostic criteria have also evolved over time, allowing earlier diagnosis for earlier treatment and prevention of disability progression in the long run. So the current scenario is characterized by early diagnosis and early treatment, availability of different therapeutic options with different safety and efficacy profiles and risk to benefit ratio. Which calls, of course, for accurate prognostic profiling of the patients in order to tailor the aggressiveness of our therapeutic choice, and of course the need to discover reliable prognostic biomarkers. Against this scenario, the main aims of the project were to identify MRI, genetic, serum, and cerebrospinal fluid biomarkers, prognostic biomarkers, in an early stage of the disease, and to identify a combination or pattern of these biomarkers that is more capable to predict subsequent disease course. As a secondary aim, also to test the capability of this pattern of biomarkers to predict treatment response. So we recruited patients with multiple sclerosis consecutively referred to our MS center who had a first assessment within 1 year from the clinical onset of the disease, so really an early stage of the disease. And our clinical assessment included the assessment of disability on this EDSS scale, and also we provided a neuropsychological and psychosocial assessment using well-validated scales for cognition, depression, and fatigue. MRI assessment was performed using a 3 Tesla scanner to measure total and regional brain volume, lesion load, spinal cord assessment, and we also looked at CSF biomarkers, particularly biomarkers of microglial activation, serum biomarkers, and especially the neurofilament light and GFP, and our genetic analysis focused on the polymorphism Val66 MET of the brain-derived neurotropic factor and the polymorphism epsilon-4 of the ApoE. So brain volumes and lesion volumes were obtained using an algorithm based on convolutional neural network. And we also assessed these slowly expanding lesions that literally are lesions that tend to increase in their volume over time and are emerging as a powerful prognostic marker of a more aggressive, more progressive disease. And notably, we were able to find these lesions in almost 60% of the patients after only 3 years after the diagnosis. We also calculated the volumes of different subfields of the hippocampus and the nuclei of the thalamus. So these are some results from our analysis. To date, we collected MRI data from 314 MS subjects and BDNF polymorphism in 193 subjects. Our analysis is focusing on the polymorphism, the BDNF polymorphism, and its relationship with the hippocampal volume and clinical progression of disability over time. So just a short focus on the hippocampus, that is, of course, an extremely important region of the brain for cognition and memory in particular, and is considered a neurogenic niche. It's a unique environment that hosts neural stem cells that can differentiate into neurons, oligodendrocytes, and astrocytes. And in multiple sclerosis, it is well documented that the hippocampus is severely damaged with severe demyelination, neuronal damage, and synaptic damage. The hippocampus plays its role through this neurotrophic factor, the BDNF, that is highly expressed in this region and has a number of key functions in neuronal survival, growth and differentiation, neuroprotection, synaptic plasticity, and modulation of neuroinflammation. So in a previous cross-sectional and small study, we found an association between the polymorphism of BDNF and the preserved volumes of the hippocampus, suggesting a potential protective role of this polymorphism. And we aimed to explore this hypothesis in a larger sample and in longitudinal study. These are the sample characteristics at baseline. We found the polymorphism in 68 patients. The characteristic clinical and demographic were similar. Only females were more represented in the non-carriers group. Also, the MRI characteristics at baseline were similar. We found, however, higher white matter volume in the non-carriers group, but importantly, again, we found a significantly higher hippocampal volume in the group carrying the polymorphism. And this is a focus on the specific subfields of the hippocampus that show a higher volume in the carriers of the polymorphism. In the longitudinal analysis, we found an interaction of the polymorphism with time, and again, consistently with cross-sectional data, we found a lower rate of progression of hippocampal atrophy in those carrying the polymorphism. These are the subfields, the subiculum body, the hippocampal amygdala transition area, the hippocampal head, and fissure. Importantly, the carriers of the polymorphism exhibited a lower rate of disability progression during the follow-up, and In conclusion, the key findings so far, and our inference from these findings, we found this association, cross-sectional association at baseline between the presence of the polymorphism and the preserved hippocampal volumes. We confirmed in a longitudinal analysis this reduced progression rate of hippocampal atrophy in the carriers of the polymorphism that was also associated with the lower rate of clinical progression of disability. And we hypothesized that this polymorphism could have a protective effect, possibly due to enhanced brain plasticity and neurogenesis in the hippocampus, thus contributing to limit the evolution of clinical disability. Of course, we need to complete the assessment of other biomarkers and eventually to run this machine learning algorithm to find out the optimal combination of predictive biomarkers. And this is my last slide, acknowledgements to Rossella Marcucci and the group of collaborators. Thank you for your attention.